Okay, good afternoon. Today we're going to talk about a very interesting series of events that occur in some virus-infected cells: reverse transcription and integration. We're going to talk about two virus families, the retroviruses, which do both of these, and the Hepatitis B viruses, the hepadnaviruses, which just do reverse transcription. This story begins in 1908, just not too long after viruses were first discovered. Remember, at the end of the 1800s, the first plant virus, tobacco mosaic virus, was discovered. 1908, a virus was discovered that causes leukemia in chickens. Now, at the time, no one thought leukemia was a real cancer or a real tumor, so it didn't get a lot of attention. But then, a few years later, Peyton Rouse, 1911, he was working at Rockefeller Medical Institute right here in New York. He discovered a virus that causes sarcomas of chickens, solid tumors of chickens. And that was called Rouse sarcoma virus. And we will revisit this discovery in another lecture. It got him the Nobel Prize 55 years later. It's the longest incubation period ever between the experiment and a Nobel Prize. And not too long after they were discovered, they were called tumor viruses. All right, so that's the subject of one of our topics today, tumor viruses. These two, the chicken leukemia virus and the sarcoma virus, were eventually found to have RNA genomes. And then the question arose, how could they transform cells? So we will talk about transformation later. The transformation is a change in the cell such that it's on its way to becoming oncogenic or tumorigenic. And the question was, how do these RNA viruses change a cell in such ways? They have RNA genomes and the cell is DNA. It was a puzzle. Well, that was sorted out in 1970 when two independent investigators, Howard Temin at the University of Wisconsin-Madison, David Baltimore at MIT, discovered reverse transcriptase in the virions of RNA tumor viruses or retroviruses. And they got the Nobel Prize for that 5 years later. So they discovered this enzyme, reverse transcriptase. We have mentioned it before in this course. And it was called reverse transcriptase because it reversed the flow of information, the so-called central dogma, which says that you go from DNA to RNA to protein, and you never go back the other way. So this was called reverse transcriptase because, as you will see, it takes RNA and makes a DNA copy of it. So it goes against the central dogma. So much for the central dogma. So then the viruses were called retroviruses because they use reverse transcriptase to reverse the flow of genetic information. So where do these stand among all the viruses that we have talked about so far? Remember, this is the Baltimore scheme. And this is David Baltimore's idea, this Baltimore scheme. And he's one of the co-discoverers of reverse transcriptase. So here's our mRNA in the middle. And the retroviruses we will talk about today here, they have a plus strand RNA genome, but it is not translated when it gets into the cell. It's converted to first a single-strand minus strand DNA, then a double-stranded DNA copy, which then goes to mRNA. We will also talk about the hepatitis B viruses, which have reverse transcriptase in their life cycle as well, even though you don't see it on this scheme. We'll talk about those later on today. So these are the two abstracts of these 1970 papers from Temin and Baltimore. Again, I said they independently discovered reverse transcriptase in the virions. So they were working with purified virus preparations. They found the enzyme in the virus particles. So that means that the viruses carry this enzyme into cells. And they found it by doing biochemical assays on these purified viruses. They would treat them with a little bit of detergent to make them permeable and add radioactive precursors and follow the incorporation of those into DNA. And we actually had David Baltimore on our 100th podcast as sort of a celebration, and so that's very informative if you want to learn more about that discovery. We also talked about reverse transcriptase another time. Now, Temin had this particular insight. He said not only does this enzyme, reverse transcriptase, convert the viral RNA to DNA, but then the DNA gets to be a permanent part of the host cell. And he said that is how these viruses cause tumors. And how they do so we will see later on, and it's really interesting. But this was his insight. He said the DNA of these viruses become a permanent part of the host DNA. He called it a provirus. All right, so the integrated copy of the retrovirus genome in the host cell is called a provirus. And he said this must be how these viruses transform the cells, make them transformed, which leads to the formation of a tumor. Now, this idea had already been around from studies of a bacterial virus, bacteriophage Lambda. So we have only mentioned bacteriophages briefly in this course. This is a depiction of the structure of Lambda. It's a virus with an icosahedral head, which is full of double-stranded linear DNA. It has a tail and then tail fibers through which it attaches to E. coli. And it injects this double-stranded DNA into the bacterium, and then it circularizes and replicates. And under some conditions, this DNA, this phage DNA, which is shown here as a red circle, it's double-stranded, can integrate into the E. coli genome. And it does so at a very specific site. And what it does is it linearizes the phage genome and makes it part of the E. coli genome. So E. coli genome is blue. You can see on either side of the red phage genome. So this had already been known for some time, and this undoubtedly influenced Temin's thinking because there was precedent for the integration of viral DNA into that of the host cell. So that's probably where Temin got some of his inspiration from. So now we know a lot more about retroviruses, and we'll start by looking at the particles. This is, on the left, actually an electron micrograph of Rous sarcoma virus. It's a virus that Peyton Rous discovered in 1911. On the right is a schematic of a similar retrovirus. These are enveloped viruses. They have a lipid envelope derived from the host cell. It contains viral glycoprotein, a single viral glycoprotein composed of an SU, or a surface portion, and a TM, a transmembrane portion. The TM goes through the membrane. It's non-covalently linked to the SU portion. Beneath that, there is another viral protein called the matrix. You can see it in blue here. It gives structural integrity to the envelope. And then within that, there is a capsid. So you can see here, this black structure is called the capsid. It's made up of a protein called capsid protein. And within that is the viral RNA, these two green molecules, plus stranded RNA again. It is bound to protein. It is coated with protein. The protein that coats it is called NC, or nucleocapsid. And this is unusual for a plus-strand RNA virus. As we've said before, if the RNA is destined to be translated as soon as it gets in the cell, it doesn't need to be coated with protein. This RNA doesn't have that destiny. It's going to be copied into DNA. The virion also has a few other enzymes within it. It has the reverse transcriptase right here. It has an integrase, and it also has a protease. And the roles of those will be apparent shortly. Now, this is a map of the genome of these viruses to give you a sense for how the genetic information is expressed. At the top is a map of the provirus. What that means is this is a map of the DNA as it sits in the host genome. All right, we don't have any host genome showing here, but it would be on either side. The two outermost sequences are called LTRs, long terminal repeats. We'll talk about those quite a bit. And then within is the coding region. There are a series of proteins encoding structural components like the capsid and the nucleocapsid, the matrix protein. This is all called the GAG region. Then there are enzymes, the reverse transcriptase and the integrase, and one other encoded here. The protease is in yellow right here. And then finally, the envelope glycoproteins are encoded here. Now, the way these are expressed is from two mRNAs, depending on the virus. There's one mRNA that begins in the LTR. It's capped. It goes the whole length of the provirus, and then it's polyadenylated within the other LTR here. This is translated to produce the structural proteins. There is a termination codon right here at the end of the GAG region, and that has to be overcome. About 5 to 10% of the time can be either suppressed or frame shifted. We'll talk about that next time. So you get a longer protein made. So now you get a GAG-Pol fusion, and that gives you the enzymes, the reverse transcriptase, the integrase, etc. However, the envelope glycoproteins can't be accessed from this message. What happens is a subset of these messages are spliced. These internal sequences are spliced out, and that gives the ribosomes the ability to translate these structural glycoproteins. Okay, so you have a little bit of proteolytic processing, suppression or frame shifting, and splicing to get at all of this information. Now, not every retrovirus does the same strategy. There are more complex examples, but just gives you a flavor of what happens in terms of decoding. Now let's look at the infectious cycle of a typical retrovirus. Here's a single cell. We have a virion binding to a receptor. There are many different kinds of receptors for retroviruses, depending on the specific one. Here we show the envelope fusing at the plasma membrane. For other retroviruses, they're brought in by endocytosis. So again, there's variation there. And then the core is deposited in the cytoplasm. The core contains the viral RNA genome. And you may notice there are two copies of the genome shown. We'll look at that in a moment. The RNA is reverse transcribed because remember there's the RT enzyme in the particle. It's reverse transcribed to make initially a negative single-strand DNA and eventually a double-strand DNA. All of that happens in the cytoplasm. And then this double-stranded DNA is imported into the nucleus and then integrates into host cell DNA. It's absolutely obligatory to integrate, otherwise the genome will not be expressed as mRNA. So this is the provirus now. It's an integrated DNA copy of the viral genome in the host cell. Transcription, you may recall from last time, then occurs as the provirus is sitting in the genome, and it's done by host RNA polymerase II. So host Pol II is a cellular enzyme. It recognizes a promoter in the left-hand LTR of the proviral DNA, and it makes these full-length mRNAs. And remember the previous slide, the longest mRNAs are translated to give structural proteins and enzymes. And those, of course, are translated after they're exported into the cytoplasm. The spliced mRNA, the shorter one, gives rise to the glycoproteins that are eventually exported to the plasma membrane to make new virions, and that's a topic we'll consider during assembly. So the key here is that you have production of a DNA copy, integration, and then the host cell makes mRNAs for the virus. So the only enzyme that the virus, nucleic acid-based enzyme that the virus has is reverse transcriptase. All right, let's look at some of these steps in more detail now. Let's start with reverse transcriptase. This is, again, a viral enzyme. It copies the viral RNA to produce DNA. It is a primer-dependent enzyme. The primer can be DNA or RNA, and you will see examples of both today. The primer must be completely base paired, as it's shown here. So here's an RNA template. The primer is base paired to it in its entirety. The template can be RNA, the viral RNA, or it can be DNA. As you will see, the reverse transcriptase not only makes a single-strand DNA copy, but then it makes it double-stranded. So the template can be RNA or DNA. Only deoxy-NTPs are incorporated, the precursors to DNA. This enzyme will not use ribo-NTPs, will not make RNA. But you could change the enzyme to make it do that. All you have to do is change one amino acid in this enzyme, and it will now make RNA instead of DNA. And of course, the way this works, the enzyme uses a primer, and then it extends a DNA based on the template, of course, by reading the individual bases. And so this is an enzyme you should be familiar with by now because it looks like all of the other nucleic acid polymerases that we talked about previously. Now, reverse transcriptase has a second activity. It's called RNase H. And this is an enzyme that cleaves RNA, but it has to be double-stranded. It has to be in a duplex form. It can either be RNA-RNA, which would be double-stranded RNA, or it could be an RNA-DNA hybrid. That's why I say duplex form, because it can be either one, either RNA-RNA or RNA-DNA. So what the enzyme does, it makes endonucleolytic cleavages, which means it cleaves in the interior of the nucleic acid. It doesn't just chew from the ends. That would be exonucleolytic. It cleaves endonucleolytically, and it makes short oligonucleotides with 5' phosphates and 3' hydroxyls. So here's an example of it could be an RNA or DNA RNA. Let's say this is a DNA RNA hybrid. So this would be the viral RNA, and then this would be the first strand of DNA made by reverse transcription. The RNase H is chopping up basically the RNA. It doesn't need it anymore. It's now got a DNA copy. It does so endonucleolytically. It cuts 5' of each phosphate. So this is the phosphate joining the bases on either side, a part of the backbone, 2 oxygens on either side. And this RNase H cleaves in front of each phosphate. So it gives you 5' phosphates, and then at the other end it would give you a 3' hydroxyl. It's simply a way of getting rid of the RNA because, as I said, it's not needed anymore. Right, so that's RNase H. Now, the virus particle, again shown up here in the upper left, has 2 copies of the genome. This is essential to have 2. You can't have them with 1. If there's only 1 RNA, it never gets in the virion, as we'll see later. So within the capsid is 2 RNAs, and here they are blown up here. 2 copies of plus strand RNA. Here's the 5' end. It's capped. 3' end is polyadenylated. They are associated in this region. There is some loose base pairing that occurs to keep them together, and this is actually very important for their incorporation into virions. Only these 2 molecules together can be packaged into a virion. In the virus particle, these 2 genomes, are coated with NC protein. That's why they look a little fuzzy here. Nucleocapsid protein coats the RNA. And then there are about 50 to 100 molecules of reverse transcriptase per virion, in addition to the RNA. And then, of course, these other proteins, integrase and RNase H, are also present. Now, the dimer, why is there a dimer of RNA? We think there's a dimer to help the virus be genetically resilient. It turns out that retroviruses are pretty resistant to UV irradiation, ionizing irradiation. These are treatments that introduce mutations in the genome. And retroviruses, compared to most other RNA viruses, are pretty resistant. And we think this is because there are 2 copies of the genome. So in essence, what we think happens is that when the reverse transcriptase is copying the RNA, it switches back and forth randomly between one copy and the other. So eventually you get out, if you have a mutation in one of the 2 RNAs, it's going to be fixed by the complement in the other RNA, or by the other strand of RNA. All right? And we think that happens by copy choice. This is a slide that we looked at a couple of lectures ago to illustrate RNA recombination. It shows a polymerase copying one strand of RNA and then switching to another. So if you can imagine that this is the RNA, the dimeric RNA of the retroviruses, and this is reverse transcriptase, it's constantly switching back and forth. In addition, this reverse transcriptase is really has a real poor processivity. So processivity of an enzyme is its ability to keep going. Enzymes with good processivity plow through the template and never come off. The RT goes some bases, it falls off. Another one comes and takes its place and goes on. It's happening all the time. It takes hours to copy one RNA to make a DNA. So it has poor processivity. So enzymes are copying, they fall off, another one comes on, it switches to the other strand. So we think that having the 2 genes coupled with this copying mechanism ensures an error-free DNA copy. Now let's look at the process of reverse transcription in detail. Here we have the genome at top, again, the dimeric genome, 2 copies in the virion. Let's blow it up, just the left-hand side. And you may have noticed in the previous slide there were these 2 green molecules folded up very much. These are actually tRNAs, which are also present in the virion. There are 2 tRNA molecules which are hybridized to the viral RNA. And you can see now in this blow-up of the left-hand region, we're going from the left-hand part of the mRNA, which is capped, to just past the end of this LTR-like region. And here is a tRNA base paired to the RNA. It's base paired in a region called the PBS. It's the primer binding site. This tRNA is going to be the primer for reverse transcription. Remember I said the RT is a primer-dependent enzyme. For retroviruses, the primer is a tRNA. This is a cellular tRNA that it steals from the cell when it packages the genome. Now it's shown here just hybridizing in a very straightforward manner to the primer binding site, but in reality it's more complex, and that's illustrated at the bottom. Here in this region of the primer binding site, you can see PBS is right here. This forms a duplex RNA structure. So the RNA form base pairs with various bulges and loops. And the tRNA binding site is right in here. Here's the tRNA. This is a classic tRNA folded into a 3-stem loop type structure. This tRNA base pairs, and it melts out the secondary structure around the primer binding site to form a structure like this. So now that you can see the structure of both RNAs have been changed. The tRNA has been changed by the base pairing that you can see here. Here's the 3' end of the tRNA. That's going to be the initiation site for DNA synthesis. Here's the 5' end. So you can see it forms this very invasive type of hybridization. This happens to be for a very specific retrovirus, the avian sarcosis and leukemia viruses. It uses a tryptophan tRNA. Depending on the retroviruses, they can use various different tRNAs. So that's the primer for DNA synthesis. And let's see how that happens now. And again, this all happens in the cytoplasm. By the time this gets in the nucleus, it's going to be double-stranded. So here we have on the left our viral RNA. We only show one copy now for simplicity, but it's pretty complicated. This is going to be happening with 2 copies, and the process is going to be jumping back and forth. So here's the tRNA hybridized to the primer binding site. And look where it is. It's right near the 5' end of the genome. That's pretty strange, because at best, you're going to only be able to make a short piece of DNA. And what happens is, in fact, the reverse transcriptase uses the tRNA as a primer. It makes a short piece of negative strand DNA, and then it stops because there's no more template. You'll see why this happens in a few moments, I think. Now, as the DNA is made, the RNase H activity of RT chops up the template. That's what these little bits are meant to represent. So this template is now gone. And you have still hybridized, the tRNA is hybridized to the RNA, and now you have a little piece of single-strand DNA, about 100 bases or so, negative strong stop DNA it is called, which is overhanging here. Turns out that this has complementarity to the other end, the 3' end of the viral RNA. So if you look back here, the 5' end has very specific sequences called R and U5. The 3' end has R and U3. As these stand right now, these are not LTRs, but we're going to build 2 LTRs from this in the end. And those are absolutely essential for proviral expression, and that's why we have to go through this very baroque process. So the R sequence on this strong stop DNA is going to hybridize to the little r sequence. They're complementary at the 3' end of the RNA. And that takes us to the next step, because as you can imagine, when that happens, this hybridization of the minus DNA, then the reverse transcriptase can continue to use this as a primer. So it's using a DNA primer now. And it's going to continue copying this RNA genome, and that's what you see here in blue. As the RT is copying, the template is being chopped up. So here's the green template, and now you see the green is being chopped up as the blue is moving along. And eventually the blue will get all the way back here. But at one point, there is a piece of RNA left undigested, if you will. It's called PPT, polypurine tract. It's going to serve as a primer for the plus strand of DNA right here. So that light blue strand is minus strand. It eventually goes all the way around and copies the entire genome. But while that's happening, there is a second initiation step occurring on that minus strand DNA from the PPT primer. So it's a little piece of RNA left behind as a primer. You can see it's being extended here. It's dark blue because it's now plus-stranded DNA. Okay, we're almost there. So now the minus strand is complete. It's made a complete round of the genome. It's reached the primer binding site where the tRNA was originally bound on the incoming virion. A little bit of viral RNA is left there. In the meanwhile, the polypurine tract RNA serves as a primer for plus strand, and it goes through the tRNA, the primer binding site on the tRNA, and then the tRNA eventually is cleaved off by RNase H. It is an RNA after all. Okay, now we have base pairing of this plus-stranded DNA to the primer binding site on the minus strand. It's going to go all the way around to make a complete DNA copy. So now we have this configuration. The plus-strand DNA, which initiated at the PPT RNA primer here, has now jumped, or we say template jump because it goes to the other end of the molecule, to the primer binding site. It's going to eventually go all the way around and make a plus strand. So now we have a double-stranded DNA copy of the viral RNA. And what you see here, and here's the linear version, we now have a plus and a minus strand. And at the left hand, we have U3RU5 sequences, and at the right hand, we have U3RU5. Those were not present at both ends of the viral RNA. The reason we had to do all these weird primings and jumps was to make an LTR at both ends of the proviral DNA. So you can look at the original RNA that we started with. Remember, it had R5 and U at one end and RU3 at the other end. Now we have U3RU5, U3RU5 at both ends. So that's what all that funny priming leads to. All right, so this is, again, happening in the cytoplasm. And this is in the virion. The virion never totally comes apart. It's a sort of subviral particle that this happens in. And then once we have double-stranded DNA made, that can go into the nucleus. Okay? This took about 20 years to figure out in various laboratories. And then another 20 to figure out how to draw it so that people would understand it. But the main point to get from this is that all of this funny business is to make two LTRs, because as you will see, these are absolutely essential for what's going to happen next. A few words on reverse transcriptase. This is a slide we looked at a while ago in RNA synthesis discussion. It has representations of the amino acid sequences of the 4 different kinds of nucleic acid polymerases. This is RNA-dependent RNA polymerase, which I talked about. Here's our reverse transcriptase. You can see it shares some of the features as the others. And it's like the others, its structure is that of a right hand, and the active domain is in the palm and in their fingers and thumb domains. You may recall that I said a signature for RNA-dependent RNA polymerases is a GLI-ASP-ASP sequence in the C domain. In fact, the reverse transcriptases have an ASP-ASP in this region. It's a signature for a reverse transcriptase. Structure of reverse transcriptases have been solved at high resolution by X-ray crystallography. And at the top is the structure of reverse transcriptase with its RNase H domain bound to a template and primer, an RNA DNA template primer combination. And what you can see here, there's a palm domain which has the active site right here in their fingers and thumb domains. The RNase H domain is far from the active site here. And what happens, if we look at this in a schematic way, here is a schematic of this structure. Here's the incoming RNA template in green. Here are 2 metal coordinating amino acids. We talked about the 2 metal hypothesis for nucleic acid catalysis. The RTs are no different. So there, the 2 metal ions would be chelated here and assisting in the polymerization. And then we would make a strand of DNA, which would be base paired with the template. That's why we show it as a helix here. And then here is the active site of RNase H. It also has 2 metal coordinated to 2 amino acids, 2 specific amino acids, and that is degrading the RNA, and then what you get out is a single strand of negative strand DNA. Now, although it looks like all this happens on one polymerase, in fact, because this is such a poor processivity enzyme, it doesn't go very far before it falls off. And then another enzyme takes its place and degrades some of the RNA, maybe makes some more DNA and so forth. So this idea of a very well-oiled machine chugging along, making DNA, and then digesting the RNA at the same time is not really correct, because no one enzyme stays on the template to be able to do all of that. But in principle, you see there's a separate active site for polymerization of DNA and for digesting the RNA. So this enzyme is pretty slow. It takes about 4 hours to copy an RNA genome. It's about 9 kb, these retroviral RNAs. It takes about 4 hours to copy it. And it makes a lot of mistakes. It makes an error every 10,000 to 1 million bases. All right? And that's typical of RNA polymerases in general. We'll come back to that later. Now, reverse transcript, yes?
Cellular RNases have much higher processing.
Yes, cellular polymerases tend to be more processive than this guy. I don't know why this one is so poor, but that's probably why there are 100 RTs in the virion, to make sure that in the end they get a DNA made. Reverse transcriptases are not just in retroviruses. It turns out that they're in bacteria and archaea. You can find reverse transcriptase in E. coli, myxobacteria, which are soil-dwelling bacteria, archaea. They have reverse transcriptase activity. You could break these open and do a reverse transcriptase assay, which is very simple. You take an RNA template with a primer, and you look for incorporation of DNA. And you can find it in many different sources. So we think this means, the finding of RT in these species, that it's a very old enzyme that evolved before these three forms of life separated. So here's a phylogenetic tree of life: bacteria, archaea, and eukaryote. They had a common ancestor. So reverse transcriptase is probably somewhere up here, because they all have it. And that's why I said earlier at the very beginning of the course that reverse transcriptase could be the bridge between the so-called RNA world, which may have existed first before a DNA world. So there was an RNA world where RNA had catalytic activity. It then acquired the ability to make proteins, and then it had to get enzymes to copy it. These are RNA duplicating enzymes. And then you can easily go from an RNA-dependent RNA polymerase to a reverse transcriptase with one amino acid change. And then you can make DNA. And so that becomes a reverse transcriptase. So the DNA world may have been introduced by reverse transcriptase. It's all theoretical, of course, but there's some support for this idea. All right, so now we have a double-stranded DNA copy of our retrovirus RNA. It's right here. We have an LTR on the left. We have the middle portion of the genome that codes proteins, and then there's an LTR on the right. The next thing that's going to happen is this is going to integrate into the host genome, into what we call a target sequence. This is almost random. There is no specific integration sequence. Lambda, for example, finds a very specific sequence in the E. coli chromosome, but that's not the case at all with retroviruses. It finds a host target. It will integrate into it. We're going to talk about the mechanism in a moment. The result is proviral DNA, shown here. We have host DNA. It's in purple on either side. And there are two very specific features of this integration that will tell you, in fact, if there is an integration caused by a retrovirus. First of all, the target sequence is duplicated. So you have this host target sequence in orange. It is duplicated on either side of the proviral DNA. We'll see how that happens in a moment. And the other is that there's a loss of sequence from the ends of the LTR. Here, you see two bases are lost. We have AATG, and we only have TG left. And the number of bases varies. It could be 2 or 3 or 4, depending on the retrovirus. So those are two characteristic features of retroviral-mediated integration: duplication of the target sequence and loss of some retroviral sequence at the ends. Now, when this virus is integrated, the proviral DNA is the substrate for mRNA synthesis. I think you've heard this briefly from Dr. Silverstein. There is a promoter in the left-hand LTR, a viral promoter, which is recognized by host DNA-dependent RNA polymerase, and the result is a capped and polyadenylated mRNA that spans the entire genome. And that's how the genetic information is expressed. Right? Now let's look at this integration reaction. So this is carried out by another viral enzyme called integrase. And what the integrase is shown as just an outline back here. We'll look at its structure in a moment. The retroviral DNA is recognized by integrase, as is target DNA. So here's our target host DNA. This is retroviral double-stranded DNA. First thing that happens is the integrase chews back the retroviral DNA. It takes a couple of bases off of the 3' end of the plus strand. You can see that here. Now there's a little 5' overhang over here. And it takes a few bases off of the 5' of the other strand. So now we've lost those 2 or 3 or 4 bases. These will now serve to, the integrase nicks the host target DNA at these 2 sites. You can see now we have a nicked target DNA. It chews back, it doesn't chew back, it just nicks the target DNA. So now we have 2 single-strand 5' and 3' ends on the target DNA. Those are then ligated to the viral DNA here, and then the gaps are repaired. So because you are repairing the gaps, you duplicate the host sequence. That's where the duplication comes from. Because here, when you take and make each end single-stranded of the target DNA, when you go back later and fill it in, you get a duplication of sequence. So that explains the duplication. And of course, when the integrase chews back here to make single-stranded overhangs, that's where the loss of DNA sequence occurs at the ends of the retroviral genome. So again, you have the integrase carrying out the deletion of the ends, the nicking of the target DNA, the ligation of target to the viral DNA, and then repairing the damage. So now you have a proviral sequence here. It's integrated into the genome. So let's see how that looks at a closer level. We know the structure of the integrase. It's shown here. It's a dimer of a single polypeptide. And in cartoon form, that dimer is shown here. So here is one monomer and the second. And there is a catalytic region, which is shown right here. Here is the host DNA in purple again. It's double-stranded, of course. The viral DNA has already had its ends chewed back in this structure here in this depiction. So you can see the 3' end here and here. These are attacking the phosphate bonds here. They will nick the phosphate bond and be ligated to it. And now you can see that when those are ligated to it, the next step, of course, will be the repair and the duplication of the target sequence. So this happens on the surface of the integrase, which has all these activities: the initial exonucleolytic attack on the viral DNA, the endonucleolytic cleavage of the host DNA, and ligation and repair. All of that is carried out by the integrase. So it has cleavage as well as polymerization activities. Now let's talk just a bit about the integration, because we have big genomes that are targets for retroviral integration. So does it go in any particular place, or is it specific? There is no, as I said, there is no specific sequence. If you infect cells with a retrovirus, it will integrate, and then you clone out all the integration sites and sequence them. You don't get any consensus sequence. But you do get certain preferential areas, and that's what's shown in this experiment. So we'll just look at the left here at the integration of proviral DNA of 3 different retroviruses into human DNA. So you infect human cells with 3 different retroviruses, and then you clone out all the integrated proviruses and you sequence the integration site and you ask where they are. So these are 3 different retroviruses. We have a sarcoma leukosis virus, it's a chicken retrovirus, a murine leukemia virus, and an HIV, a human retrovirus. And these are the percent integration either within genes or at transcription start sites. So the reason this was done is because the transcription start site tends to be unwound in the chromatin structure and accessible to the polymerase. So the question is this a good place for retroviruses to integrate, because it is accessible? Now, if you just do a mathematical calculation based on the number of genes or the number of transcription start sites, you get these numbers. If your provirus was integrating randomly, 26% of the time it would be within a gene, 5% of the time would be within a transcription start site. It's just based on the number of each of these elements in the genome. And you can see that this retrovirus, for example, has almost a twofold increase in integration within genes compared to random. So there's some preference for integrating within genes. And you can see it's about the same for transcription start sites as random. So there doesn't seem to be any particular preference of this one. But look, the mouse leukemia virus, it certainly has a preference over randomness of transcription start sites. It also has a preference for within genes, and HIV as well. So there is a preference for, in certain retroviruses, for sequences that are being, and in fact, unwound by being wrapped around a nucleosome, as well as in some cases transcription start sites. But there is no specific sequence that you could say this is a retrovirus integration site. It can go pretty much anywhere within these constraints. So there again is our proviral DNA. It's integrated into host DNA. The duplication of the host DNA is again shown in orange. And this is now the substrate for transcription, for production of mRNA. Remember, the goal is to get to mRNA so you can make protein. The LTRs have a promoter built into it, which is built during reverse transcription. The RNA, the viral RNA, doesn't have an LTR. But at the end of this reverse transcription, when we get this double-stranded 2-LTR product, you have a promoter in the left-hand LTR, and you have a polyadenylation site in the right-hand LTR. And the product of that is this mRNA, which again is made by host DNA-dependent RNA polymerase. So this virus depends entirely on the host to make its message. The only thing it does is to make a DNA copy. And as you will see, these messages, some of these messages will be used to make new virus particles. So there's really no DNA replication because the virus makes a DNA copy from one input RNA or two input RNAs. It doesn't replicate its DNA, and it doesn't replicate its RNA. The host does that. It's very curious, in fact, that we don't have these DNA-dependent DNA polymerases or RNA polymerases replicating the genome. So that brings us to this part. Here's the provirus again, integrated into host DNA. The promoter in the left-hand LTR allows production of these mRNAs, again, entirely by host DNA polymerase. Those are all exported out of the nucleus to give rise to proteins. And as I said before, we'll talk about these later steps in virus production in a couple of lectures. Now, this proviral DNA becomes a permanent part of the host chromosomal DNA. There's no way to get it out. The virus has no mechanism for getting rid of this proviral DNA. It doesn't want to. It's happy to be there because the host is churning out mRNAs, which are going to be new viral genomes. If a skin cell were infected with a retrovirus, the DNA within that skin cell would contain proviral DNA, but eventually the skin cells would go away, they would die or fall off, and the DNA would be gone. However, some retrovirus can infect germ cells, and then they become a permanent part of your lineage. We call these endogenous retroviruses. It's a specific term meaning that the provirus is in germ cell DNA. So it is now passed along as a gene by Mendelian rules. And in fact, just about every living thing that is infected with a retrovirus, their genomes are full of these, what we call retroelements, remnants of retroviruses, proviral DNA. Some of them are active. So wild mice, for example, have endogenous retroviruses in their genomes. They pass them from mother to children, and they actually make viruses. So if you go out and catch a wild mouse, that wild mouse is making retrovirus particles. We have endogenous retroviruses in our genomes, but they're all dead. They're all extensively mutated, so we don't make any infectious retroviruses. So depending on when in history this has happened, they may or may not be infectious. You can use this as a way of dating the point of insertion. So you can do phylogenetic trees of speciation from humans to pre-humans and so forth. And these endogenous retroviruses typically are at one or a few places in the genome, and that's conserved as far back as you look in phylogeny. So you can use this to estimate when the original infection occurred. And you can also, by that, estimate how old viruses are. And that's how some of the numbers we get about the age of viruses come from. So the presence of these sequences in the genome are called, in general, retroelements because they are derived from retroviral infections, and the sequences resemble retroviral sequences. All right, retrovirus-related sequences, and as I said, they're endogenous when they're in the germline. They're often replication defective. Our endogenous retroviruses are all replication defective. Half of our genome, half of our DNA is retroelement stuff, all right? Which is not only endogenous retroviruses, but as you'll see in a moment, other kinds of sequence elements related to proviral DNA. It's amazing. So we estimate that they have been there for a long time, certainly way before Homo sapiens evolved, to precursors of hominids, and maybe even before, at least 1 million years ago. So at some time, a long time ago, one of our precursors got infected with a retrovirus. It went into the germline, and it has stayed in that position all these years. It's accumulated mutations. Maybe initially it was making virus, but over the years it got mutated, no longer makes virus. So today we don't make any virus, but it's been there a long time. So these are the different kinds of retroelements that you can find in eukaryotic genomes. And as I said, there are a lot of these. Half of our genome is composed of this stuff. So at the top is an endogenous retrovirus. This is a retroviral, proviral DNA integrated into the genome. Here is host cell DNA in purple. The arrows show you the duplication of host cell DNA that occurs when a retrovirus is inserted. So if you sequence these elements in genomes, so right now people are sequencing all kinds of genomes of living things, and you can scan them for these kind of elements. And one of the key telling points that it's a retroviral-derived element is that you have this duplication of host DNA on either side right here. So that's what that is. So here we have our LTRs, and then we have the GAG and PAL and M genes. This is a retroviral, proviral DNA. So we call that an endogenous retrovirus. We have between 1 and 100 of them in our genome, endogenous retroviruses, but they're all extensively mutated so they don't make particles. But they do look like this. Then there's another class called retrotransposons. These have very similar features, a duplication of host DNA. They have LTRs, they have GAG and PAL, but they don't have envelope genes. They've been deleted, so they don't make particles. But they do have reverse transcriptase, and they can jump around within the genome because of that, by the reverse transcriptase. So this can be transcribed. There are Pol2 promoters in the LTR, so you can make an RNA, and this can get reverse transcribed into DNA. So you have a DNA copy of this, it can jump around the genome. And this is probably how these have amplified in eukaryotic genomes over the years. So in yeast, for example, there are between 100 and 10,000 of these in the yeast genome. Then we have a variety of other kinds of retroelements. These retroposons are integrated into our DNA, accompanied by duplication. They do encode reverse transcriptase, but there's no structural proteins of the virus at all in here. We have between 10,000 and 100,000 of these in our genomes. Because they encode reverse transcriptase, they can jump around. And sometimes when these do jump around, they integrate into a gene and they cause a mutation. There's been some documented cases of certain genetic diseases which are mediated by this kind of integration. We also have another version which is called a retrosequence. Again, it has less retroviral DNA, but it's again got the characteristic flanking duplications. There's no reverse transcriptase in these. And there are about 100,000 to 1 million of these in our genome. But clearly retroviral derived. You can see by the blue sequence, it means it's got homology to retroviruses. And then finally, the last one is called a processed pseudo gene. This is basically a DNA copy of an mRNA that was probably at one point an mRNA in the cell. It got copied by a reverse transcriptase made by one of these other retroelements and then integrated into the genome, but not in an integrase H-mediated fashion. So we don't have the DNA duplication at either end. And that kind of DNA integration can happen. If you take any kind of DNA and put it into a cell, the DNA will get in the nucleus. It will integrate into chromosomal DNA without integrase, because there are all kinds of breaks that occur in chromosomal DNA, and they get ligated and repaired. So you can have this kind of non-integrase H integration. So those are retro elements that comprise half of our genome. And it's interesting that they're still there. Some people think that they might have a function, but we're not really sure, because you can't do the experiment in any animal. Even mouse has too many of these to take them all out and ask what would happen. Now, this is a phylogenetic tree of most of the known retro elements. And they've been put into three classes: class 1, class 2, and class 3. The reason I show you this is because it just gives you a sense of how many there are. These are classified according to species. The blue squares are primates, including humans. Then we have cats and rodents, ungulates, bird, reptiles, fish, amphibians, insects. Everything has endogenous retro elements in them. And also on this graph, you can see these are real retroviruses here, like HIV and other retroviruses as well. So these are replication competent, just to show you how they relate. They're highly related to the others, but of course they're on their own branch, because these are now evolving in genomes and they have no constraints to maintain viral functions any longer. But among these are some human endogenous retroviruses. They're called HERVs, human endogenous retrovirus. And as I said, we have between 1 and 100 of these in our genome. And very recently, someone repaired all the mutations in one of these endogenous retroviruses. So this is HERV-K. This is probably the most recent endogenous retrovirus that went into us about 1 million years ago. Obviously not Homo sapiens, but our ancestors. By the sequence, you can estimate that it infected a precursor of Homo sapiens about 1 million years ago. So it's the least genetically mutated of all the endogenous retroviruses that we had. So this group here in New York City, actually, at the Rockefeller, they cloned the proviral DNA out of the genome, and they fixed all the mutations so they could reconstitute a virus from it. And they made it, they put it in cells, and the cells made virus particles, which you can see here. So this is like a resuscitated retrovirus. It's been dead all of these years, and now they have it, and they can study it in the laboratory. So I find this intriguing, that you can get something that hasn't been infectious for maybe 1 million years, and you can work with it again. Now, this whole process of getting a retrovirus in your germline, that's called endogenization. That's right, they're endogenous retroviruses. That means in your germline. The process of getting the virus in your germline is called endogenization. So with people, we were endogenized millions of years ago in our ancestors, not even Homo sapiens. So someone got a retrovirus infection, it went into their germline, and then all their offspring had that retrovirus in their germline. So there's very little opportunity to watch this actually happening, although it's now happening in koalas. So koalas are native to Australia, and they are infected with a koala retrovirus. The reason why this was discovered is because the koalas, many of which are in zoos, started getting a leukemia, and so they looked into this and they found that they had high levels of viremia of this koala retrovirus. It turns out that this koala retrovirus came from a mouse many years ago. But not every koala in the world has been endogenized by this retrovirus. So there are 62 in Japanese zoos. So all the koalas come from Australia. Many years ago, they wiped out all the koalas in Australia, and there were only a few left on an island, Kangaroo Island, and then they repopulated the world's koalas from this island. The island is free of this koala retrovirus. But when they put the koalas on the mainland, they get infected. And then when they ship them up to Japan, many of those are infected as well. And so people are studying this. You can capture koalas and take some blood from them, and you can see there are uninfected koalas in the wild and there are infected ones, and you can track the spread throughout Australia. And as this virus infects them, it goes into their germline. So there are different groups of koalas with different integration sites in their germ cells. So you can watch the process of endogenization in this species. So presumably, if you're not careful, eventually all the koalas will be infected and will have this. But it's the only example I know of where you can actually watch the virus go in the germ cell and see how it spreads. Now, it could be that somewhere on the planet, someone who has HIV has been endogenized, which means that the DNA, the proviral DNA, has gone into their germline. We don't know because we don't look for it. Of all the 30 to 40 million people that are infected with HIV, we don't actually look for endogenization. But it could be that one person or several people are endogenized, and then in a few thousand years we'll see the result of that, because that takes about that much time for a gene to populate through humanity. So it's a very interesting subject. All right, so now the next virus that does reverse transcription is Hepatitis B virus. And this is weird because this is a DNA virus. Remember, we talked about this a little before. It's an envelope DNA virus, and the genome is a partially double-stranded DNA with very strange features. It has a protein linked to it and an RNA as well. We're going to see how that arises today. So these are Hepatitis B virions, and this is the genome and its coding potential. But what I would like to talk about is the replication cycle. So here's the virus infecting a cell. The genome is deposited in the cytoplasm. It gets into the nucleus, where it then gets repaired. The gaps are filled in, the protein is removed, and the RNA is removed. Once it's repaired, then it can be transcribed by host DNA polymerase to make mRNAs. The mRNAs are shipped out into the cytoplasm. They give rise to proteins. And the proteins start to build new capsids. And some of the full-length mRNAs get encapsulated. Okay? So the full-length mRNAs, like the retroviral mRNAs, get into the capsid. The difference is that the hepatitis B viruses convert them to DNA in the capsid. And they do that using reverse transcriptase. So you could almost look at this as a retrovirus that couldn't wait to do reverse transcription. It couldn't wait till the next infected cell. As soon as there's RNA in the capsid, it starts reverse transcribing it and making a DNA copy. It makes a single-stranded DNA, then a double-stranded DNA, and eventually that matures, and those are new particles. You see here, sometimes these get recycled back in the nucleus to make more closed circular DNA. But at some point, an infection gets an envelope, and now you have this DNA virus. So a DNA virus, a retrovirus that could not wait. That's basically Hep B. So how does this happen? The Hep B genome also has a priming requirement for reverse transcription, but it is not a nucleic acid primer. It's a protein primer. This is a schematic of the RNA, the Hep B RNA, 5' end here. It's capped. Here's the 3' end. It's polyadenylated. At either end, there's this very interesting secondary structure, and that's going to be the initiation site, and you'll see in a moment, for reverse transcription. So here's the full-length Hep B RNA. Remember, this is only found in infected cells. You don't find this in virions like retroviral mRNAs. Here's the 5' end. Here's the 3' end in those two structures. The reverse transcriptase of Hep B is called the P protein, right here. It itself is the primer for reverse transcription. It doesn't need a nucleic acid primer. It sits on this stem loop structure near the 5' end and initiates the synthesis of a negative strand DNA. Shortly after that is made, it hybridizes to the 3' end of the mRNA right up here. So it does a template exchange, very much like the jump of the short strand DNA in retroviral DNA synthesis. And that gets extended all around the genome to make a negative strand DNA. And at the same time, the RNA template is removed by the RNase activity of the reverse transcriptase. So now you have a negative strand DNA copy of the genome. There is now an RNA primer left at the 3' end shown here, which will serve as a primer to make the plus strand. So here's our RNA primer. It actually does another template jump. It jumps up here. So now it's priming up here. And then there's a terminal redundancy. It reaches the P protein binding site here. It can't go any farther, but it turns out it can hybridize to the 3' end of the minus strand. So it does that and ends up making a plus strand DNA. Now, there are two things I want to point out here. When this RNA primer makes this jump, it does so because its 3' end is disrupted from base pairing here by the formation of this stem loop here in the negative strand DNA. So here it's nicely base paired. All of a sudden the negative strand DNA base pairs and forms this stem loop. It bumps the 3' end off, and that allows it to find its complementary sequence up here in the other part of the genome. So here's what the genome looks like in the particles. So the negative strand is complete, the negative DNA strand is complete, the plus is about halfway. So basically what happens here, this plus strand DNA only gets about halfway around and then it stops. So the minus strand is complete, plus strand stops. We don't know why it stops, we have no idea. So that's what gives you the gap. The P protein is the protein left on the DNA, that's what's in the virion. And then a little bit of primer is left as well, and that's what that piece of RNA is in the genome. So again, this is the odd genome of Hep B that we talked about with this protein and the RNA and the gap. Now you know why it's a product of reverse transcription. All right, so this is a DNA virus, puts DNA in the genome, but it uses reverse transcription to make that DNA copy during its life cycle. So now let's compare Hep B with retroviruses. So what's boxed here is shown what is in the virion. So hepadnaviruses put DNA in the virion. It gets into cells, it's repaired, the DNA is used to make RNA, and then the RNA is reverse transcribed to make more genomes. So the DNA is what's packaged. So remember, the RNA is packaged in the cell, but it's immediately transcribed. It's immediately reverse transcribed to DNA. Whereas if it were a retrovirus, the genome is RNA. What's in the virion is RNA. It gets into a cell. It's converted to DNA. The DNA integrates as a provirus, and then more RNA is made from the proviral copy. So here's what's packaged in the retrovirus. If this were Hep B, it would package RNA, but then it would convert it to DNA right away. So that's really the main difference between the two, where the conversion from RNA to DNA occurs. In Hep B, it's in the original cell where the virion is made. In the retrovirus, it's in the next infected cell. There are two other kinds of viruses shown here that also have reverse transcriptase in their life cycles. One of them is a plant virus. It infects cauliflower. It's called Cauliflower Mosaic virus. The viral genome is DNA, double-stranded DNA, with gaps and pieces of RNA, just like Hep B. This gets into cells, the DNA is repaired, RNA is made by cellular enzymes, and then the RNA is packaged and converted to DNA in the particle, just like hepatitis B. So this is basically a hepatitis B virus of plants. Same strategy. And one more family of viruses that uses reverse transcriptase is actually a retrovirus. It's a different set of viruses within the retrovirus family. These are called Foami Retroviruses. These have a very similar life cycle to these retroviruses. They haven't, except that they package DNA, not RNA. So these Foami retros have double-stranded DNA in their virions. It gets into cells. It integrates. First thing it does, it goes to the nucleus and integrates, make more RNA. The RNA gets packaged. But before it leaves cells, it's copied to DNA. So it's almost like the Hep B strategy, where the retrovirus can't wait to start reverse transcribing, so it does so in the particle before it leaves the cell. So you can see all of these are highly related. It's just a matter of when they do their DNA synthesis. The reverse transcriptase goes from RNA to DNA in all of them, and where that happens is what distinguishes what gets in the virion, whether it's RNA or DNA. Okay. Now the hepatitis B virus, the cauliflower mosaic virus, they do not make proviruses. Their DNAs never integrate into the genome as part of the life cycle. They don't need to. They're transcribed as circular DNAs in the nucleus. The retroviruses have to integrate because that gives them a linear genome that can be transcribed by Pol2. But those aren't the only endogenous DNAs that we find in our cells, endogenous viral DNAs. as we have been sequencing genomes of animals and plants and other forms of life, we're finding sequences from a variety of different viruses in us and other species as well. And these are just some of them. So these are endogenous viruses, but they're not retroviral. We have circoviruses, they have single-stranded DNAs, parvoviruses. Even Hep B sequences have been found. Even though Hep B never integrates as part of its life cycle, we can find pieces of Hep D DNA integrated in various animal genomes. Bornaviruses, which are negative-strand RNA viruses, flaviviruses, plus-strand RNA viruses, reoviruses, influenza, VSV. These are just some of them. As we sequence, we get all this data. It's very easy to scan it, and we can find other viral sequences integrated into them. Now, these are just pieces. They are not complete genomes, and they are not obligatory parts of the viral life cycle. They are just there by accident. So, a Hep B, the DNA is in the nucleus. If it gets broken, a piece of it could get integrated into the genome. What about these RNA viruses, though? Why would a DNA copy of them be in the nucleus? Because all of these RNA viruses, they don't have any DNA phase in their life cycles, right? So the Hep B does. The DNA viruses have DNA phases, so you can imagine that integrating. But these RNA viruses, how are they getting integrated? Well, remember, most cells have endogenous retroviruses with active RT. So most cells are making a little bit of reverse transcriptase. Influenza virus replicates its RNA in the nucleus. So maybe sometimes the RNAs get copied by reverse transcriptase. You get a DNA copy made, and that can integrate randomly into the chromosome. Same for some of these other viruses. They don't actually replicate in the nucleus, but we think that cells are pretty loose with respect to compartmentalization. Just because there's a nucleus doesn't mean that things can't get into it now and then. So viruses that replicate in the cytoplasm, like reovirus, probably it's a message of Reo gets into the nucleus now and then. It could be transcribed it can be copied into DNA by RT. So that's what we think happens. You get random accidents. The nucleic acid's in the nucleus. It's converted to DNA and it integrates. Why am I telling you this? You can use this to figure out how old viruses are. So you can go back in phylogenetic trees as long as you can find these integration sequences, and you can say this virus infected this species X million years ago. So this has been done with Hep B. So they found first Hep B, a piece of Hep B DNA in the genome of all things, a finch, one of Darwin's birds. This is prophetic. This is a perfect realization. And then they could look at the phylogenetic tree of finches and find this DNA and all of it and go back and say this went in there like 40 million years ago into the finch genome. And with all of these, you can do similar things with different species. So once you find one, you look at the same place in another species that is related to it phylogenetically, and you can track it back. And so this has allowed us to go back as far as 140 million years and say these viruses were circulating back then. So you can date viruses. The other question is, do these have any function? Some of them have been shown to be transcribed by host polymerase into mRNA. Who knows? Maybe there is some positive selection for some of these. So this is a pretty new area that we're just discovering as we sequence, but could have some broader implications in biology. All right, that's all for today.